Let's begin this section by a discussion of the supportive cells of the nervous system, or the neuroglial cells. There are about 1 trillion neurons in your nervous system. That's about 10 times as many neurons in your body than there are stars in our galaxy. It's a lot. However, the neuroglia, or the supporting cells, outnumber the neurons in your nervous system by as many as 50 to 1. Neuroglia, or glial cells, are involved in supporting and protecting the neurons in the nervous system. They bind neurons together and form a framework for the nervous tissue, hence their name. Glia literally means glue. In the fetus, the neuroglial cells are involved in guiding migrating neurons to their destinations. For example, the nerves from the eyes need to traverse all the way to the back of the cerebrum into the optic cortex. How do they know where to go? Well, they know where to go because they're guided there by neuroglial cells. If a mature neuron is not in synaptic contact with another neuron, it's covered by glial cells, and this prevents the neurons from touching each other, and thus gives precision to the conduction pathways. There are 6 types of neuroglial cells. 4 of them occur only in the central nervous system, and 2 occur in the peripheral nervous system. First, there are oligodendrocytes. Oligodendrocytes are the cells that form the myelin sheaths in the central nervous system. Different cells form it in the peripheral nervous system. They have many arm-like processes, and these processes extend and wrap around a nerve fiber and form an insulating layer of myelin, which speeds up signal conduction. You'll notice that each oligodendrocyte reaches out to multiple different neurons, and it takes the interaction of many different oligodendrocytes to completely cover the axon of central nervous system neurons. Ependymal cells are cells that line the internal cavities of the brain. You can see them here. They're very similar to cuboidal epithelium, except they don't have a basement membrane, and they have these little processes that project into the material around them. The ependymal cells produce cerebral spinal fluid. This is a clear liquid that bathes the central nervous system and fills all the internal cavities. They also have patches of cilia on their apical surfaces. That helps circulate the cerebrospinal fluid. We'll consider these cells in more detail later in the semester. The next type of cell we'll consider are the microglia. Microglia are like small wandering macrophages. They wander around and clean up cellular debris and phagocytize it. These macrophages are formed from white blood cells called monocytes. It's thought that they perform a complete checkup of the brain tissue several times a day. The astrocytes are the most abundant glial cells in the central nervous system. They cover the entire brain surface and most of the non-synaptic regions of the neurons in the gray matter of the central nervous system. They have very diverse functions. The astrocytes form a supportive framework for all of the nervous tissue. They have extensions. Here, perivascular feet. These contact the blood capillaries and form a tight seal. This is the blood-brain barrier. These perivascular feet isolates the blood from the brain tissue and limits which substances are able to get into the brain cells. Thus, they protect the nervous system from many of the invading pathogens that we might find in the blood. Astrocytes also convert blood glucose into lactate, which is used to supply the neurons with food. They secrete nerve growth factors that promote neuronal growth and synapse formation. It's growth factors that would help the neurons from the eyes reach the occipital cortex, where we see the visual processing happen. Astrocytes also communicate electrically with neurons and may influence the synaptic signaling that occurs between them. They regulate the chemical composition of tissue fluid by absorbing excess neurotransmitters and ions. When neurons are damaged, the astrocytes formed hardened scar tissue and fill the space that was formerly occupied by neurons. This process is called astrocytosis or sclerosis. Now, there are only two types of neuroglial cells in the peripheral nervous system. There are Schwann cells and satellite cells. The Schwann cells are the cells that form the myelin sheaths around neurons in the peripheral nervous system. They wind repeatedly around the nerve fiber in a very similar way as the oligodendrocytes do in the central nervous system. However, there are some distinct differences that we'll address shortly. Schwann cells are also involved in assisting the regeneration of damaged fibers, which can happen in the peripheral nervous system, but not so much in the central nervous system. Satellite cells are the cells that surround the neurosoma in some of the ganglia. Ganglia are associations of cell bodies. They provide electrical insulation around the soma and regulate the chemical environment of the neurons so that one cell body isn't directly influencing the cell body of another neuron. So now we know of two different types of cells that are responsible for myelination. Myelin is the insulating layer around nerve fibers that conduct signals for long distance. It's very much like the plastic covering over the electrical wires that we see in our house. It allows long distance conduction. Remember, oligodendrocytes are the cells that form the myelin sheath in the central nervous system, and Schwann cells are the cells that form the myelin sheath in the peripheral nervous system. These myelin sheaths consist of the plasma membrane of glial cells wrapped around and around and around the axon of the neuron. That makes them composed about 80% of phospholipids, or lipids, and 20% protein. Myelination is the production of the myelin sheath, and it begins about the 14th week of fetal development, proceeds very rapidly during infancy, and is completed by late adolescence. This is one reason that dietary fat is incredibly important to development of the nervous system. In the peripheral nervous system, we'll see the Schwann cells spiraling repeatedly around. The key here is a single nerve fiber. They lay down as many as 100 different layers, wrapping and wrapping and wrapping around the nerve fiber. There's no cytoplasm between the membranes of the inner layers because they're all squished together. However, in the outer layer, or the neurolemma, we'll see the nucleus and organelles and most of the cytoplasm of the Schwann cells. Right outside the neurolemma, or the outer layer of Schwann cell, is a thin layer of fibrous connective tissue and a basal lamina. This is called the endoneurium. Now, in the central nervous system, things happen a little differently. You'll remember the oligodendrocytes. The oligodendrocytes reach out and myelinate several different nerve fibers in its immediate vicinity, but still several nerve fibers. Same scenario. It wraps around and around and around the nerve fiber. However, the cell body and most of the cytoplasm is not wrapped around the nerve fiber, forming a neurolemma. We'll also see that the wrapping occurs in a different way. They need to push new layers of myelin in and under the older ones, so the myelin spirals inward towards the nerve fiber. So again, to review some of the distinct differences. In the peripheral nervous system, Schwann cells will wrap only a single nerve fiber. And because the cell body is located in the most outer layer and most of the cytoplasm is there, we have a neurolemma. Also, the spiraling occurs with newer layers being laid down on top of older layers. In the central nervous system, the oligodendrocytes wrap around several different nerve fibers. They have no neurolemma because the cell body is located away from the fibers. And the spiraling of the layers of myelin will go inwards under the older layers towards the center and the nerve fiber. Many Schwann cells or oligodendrocytes are needed to cover one fiber. The myelin sheath is thus segmented. This neuron is a peripheral neuron, so each of these sections is taken care of by one Schwann cell. Between each Schwann cell are spaces called nodes of Ranvier. The Schwann cells themselves cover an area called the internode. That's the myelin-covered segment of the nerve fiber. The initial segment is a very short section of nerve fiber between the axon hillock and the first glial cell. This is where we'll also find the trigger zone. This plays an important role in initiating the nerve signal. Now, there are several diseases of the myelin sheath that you may have heard of: multiple sclerosis, Tay-Sachs disease. There's a little bit of information here. I'm not going to go into too much detail. However, if one of you wanted to take this on as your final project for this semester, it would be a great topic. Many nerve fibers in the CNS and PNS are unmyelinated. In the PNS, even the unmyelinated fibers are still enveloped by Schwann cells. It's a different arrangement, but they still have connection with Schwann cells. In this case, each Schwann cell holds 1 to 5 small nerve fibers in the grooves on its surface. The membrane folds once around each fiber, just overlapping itself along the edges. This wrapping is called the neurilemma, also called the mesoaxon, in unmyelinated nerve fibers. Most nerve fibers travel through individual channels in the Schwann cell, but small fibers are sometimes bundled together in a single channel. The basal lamina surrounds the entire Schwann cell along with the nerve fibers. So this is a great time to pull out a pen, close your books, and differentiate between oligodendrocytes of the central nervous system and Schwann cells of the peripheral nervous system. Quickly, what are some differences? Put it in your notes, close your books. Don't cheat until you've asked the brain to recall. Once it has a curiosity, fill in the gaps that you're missing. Open the book. What are the big differences? Re-listen to the lecture. Make sure you have it all. The conduction speed of various nerve fibers is different. It depends on 2 factors: the diameter of the fiber, as well as the presence or absence of myelin. It occurs along the surface of the fiber, not through the middle, so right along the cell membrane. Larger fibers have more surface area and conduct signal more rapidly. Myelin, however, further speeds the signal conduction. When we observe small, unmyelinated fibers, we'll see that the signal travels at 0.5 to 2 meters per second. The signal speed in small myelinated fibers increases from 3 to 15 meters per second. When we look at large myelinated fibers, the signal can travel at up to 120 meters per second. The slow signals supply the stomach and dilate pupils, where speed is less of an issue, and fast signals will supply things like skeletal muscle and transport sensory signals for vision and balance, where the signal needs to be incredibly fast. Nerve fibers in the peripheral nervous system are vulnerable to cuts, crushing injuries, and other sorts of trauma. A damaged peripheral nerve may regenerate, but only if its soma is intact and at least some of the neurilemma remains. If there's no neurilemma, then there's basically no tube for the nerve to regrow through. When a nerve fiber is cut, the distal end of the injury can't survive because it's incapable of protein synthesis. The protein synthesizing organelles are limited to the soma, so the distal fiber will degenerate, and as it does, so do the Schwann cells. Macrophages will then come along and clean up the site of injury. The soma will exhibit a number of different abnormalities when it's cut off from its target cell. It can no longer receive nerve growth factor from the target cells. The soma will swell and the endoplasmic reticulum will break up, so the Nissl bodies disperse, and the nucleus will move off-center. Not all damaged cells will survive. Some of them will just die at this stage, but often the axon stump will sprout new processes. As the severed distal end continues to show degeneration of its axon as well as Schwann cells, muscle fibers deprived of their nerve supply exhibit a shrinkage called denervation atrophy. Near the injury, the Schwann cells and the basal lamina and the neurolemma will form the regeneration tube. The regeneration tube acts to guide the growing sprouts back towards the original target. Once it's contacted the original target, the nucleus returns to its normal shape as it's receiving all the growth factors that it needs, and the nerve fiber will sometimes begin to regenerate through the tube towards the target tissue. Regeneration's not perfect. Some nerve fibers connect to the wrong muscle fibers or never find a muscle fiber at all, and some damaged motor neurons can simply die. Nerve injury is therefore often followed by some degree of functional deficit. The damaged nerve fibers in the central nervous system theoretically cannot regenerate at all. But since the central nervous system is enclosed in bones, it suffers much less trauma than the peripheral nervous system. There is information recently, though, to suggest that regeneration can occur in the central nervous system under certain situations. If you wanted to take on this as a final project this semester for presentation, that would be something interesting. Let's see what's going on new and current in the field of central nervous system neural regeneration, spinal cord injuries.